Cell structure, part 2 of 2. We will now conclude the introduction to living cells. What organelles help make and transport proteins? Proteins are assembled on ribosomes. Proteins made on rough endoplasmic reticulum include those that will be released or secreted from the cell, as well as many membrane proteins and proteins destined for lysosomes and other specialized locations within the cell. The Golgi apparatus modifies, sorts, and packages proteins and other materials from the endoplasmic reticulum for storage in the cell or release outside the cell. Cells need to build new molecules all the time, especially proteins, which catalyze chemical reactions and make up important structures in the cell. Because proteins carry out so many of the essential functions of living cells, a big part of the cell is devoted to their production and distribution. Proteins are synthesized on ribosomes, sometimes in association with the rough endoplasmic reticulum in eukaryotes. Ribosomes are small particles of RNA and protein found throughout the cytoplasm in all cells. Ribosomes produce proteins by following coded instructions that come from DNA. Each ribosome is like a small machine in a factory, turning out proteins in orders that come from the DNA boss. So, number one, proteins are assembled on ribosomes. Number two, proteins targeted for export to the cell membrane or to specialized locations within the cell complete their assembly on ribosomes bound to the rough endoplasmic reticulum. So when ribosomes are on the endoplasmic reticulum, then it's called rough. If there's none, it's smooth. When they break off, it's a vesicle. Eukaryotic cells contain an internal membrane system called endoplasmic reticulum, or ER for short. The endoplasmic reticulum is where lipid components of the cell membrane are assembled, along with proteins and other materials that are exported from the cell. Here's the animal cell. Here's rough endoplasmic reticulum, and then smooth. Endoplasmic reticulum, the portion of the endoplasmic reticulum involved in the synthesis of protein is called the rough endoplasmic reticulum, or rough ER. It is given this name because of the ribosomes found on its surface. Newly made proteins leave these ribosomes and are inserted into the rough endoplasmic reticulum, where they may be chemically modified. So the rough endoplasmic reticulum with ribosomes, proteins are assembled on the ribosomes, and the proteins targeted for export in the cell membrane or to specialized locations within the cell complete the assembly of the ribosomes bound to the rough endoplasmic reticulum. Newly assembled proteins are carried from the rough endoplasmic reticulum to the Golgi apparatus in vesicles. The other portion of the endoplasmic reticulum is known as smooth endoplasmic reticulum, smooth ER, because ribosomes are not found on its surface. In many cells, the smooth ER contains collections of enzymes that perform specialized tasks, including the synthesis of membrane lipids and the detoxification of drugs. Proteins produced in the rough endoplasmic reticulum move next into the Golgi apparatus, which appears as a stack of flattened membranes. Here's the Golgi apparatus. The proteins are bundled into tiny vesicles that bud from the ER and carry them to the Golgi apparatus. Here's the bud vesicle traveling to the Golgi apparatus. The Golgi apparatus modifies, sorts, and packages proteins and other materials from the ER for storage in the cell or release outside the cell. It is somewhat like a customization shop where the finishing touches are put on proteins before they're ready to leave the factory, modified and then sent outside the cell or kept in the cell. From the Golgi apparatus, proteins are shipped to their final destination inside or outside the cell. What are the functions of chloroplasts and mitochondria? Chloroplasts capture the energy from the sun and convert it to food that contains chemical energy in a process called photosynthesis. Mitochondria convert the chemical energy stored in food into compounds that are more convenient for the cells to use. All living things require a source of energy. Most cells are powered by food molecules that are built using energy from the sun. Chloroplasts and mitochondria are both involved in energy conversion process within the cell. Mitochondria and chloroplasts. Plants and some other organisms contain chloroplasts. Chloroplasts are the biological equivalents of solar power panels. They capture the energy from the sunlight and convert it into food that contains chemical energy in a process called photosynthesis. Two membranes surround chloroplasts. Inside the organelle are large stacks of other membranes, which contain green pigment, chlorophyll. The cellular solar plants, chloroplasts found in plants and some other organisms, such as algae, convert energy from the sun into chemical energy that is stored in food. Nearly all eukaryotic cells, including plants, contain mitochondria. Mitochondria are the power plants of the cell. They convert the chemical energy stored in food into compounds that are more convenient for the cell to use. These cellular power plants, the mitochondria, convert chemical energy stored in food into a form that can be used easily by the cell. These little mitochondria. Two membranes, the outer membrane and the inner membrane, enclose mitochondria. The inner membrane is folded up inside the organelle. The mitochondria convert chemical energy stored in food into a form that can be used easily by the cell. One of the most interesting aspects of mitochondria is the way in which they are inherited. In humans, all or nearly all of our mitochondria come from the cytoplasm of the ovum or the egg cell. You get your mitochondria from your mom. Chloroplasts and mitochondria contain their own genetic information in the form of small DNA molecules. The endosymbiotic theory proposes that chloroplasts and mitochondria may have combined from independent microorganisms. So, what is the function of the cell membrane? The cell membrane regulates what enters and leaves the cell and also supports and protects the cell. It is a working factor that has walls to protect it from the environment outside and also to serve as a barrier that keeps its products safe and secure until they are ready to be shipped out. Cell wall is made of a lipid bilayer. The composition of nearly all cell membranes is a double layered sheet called a lipid bilayer, which gives cell membranes a flexible structure and forms a strong barrier between the cell and its surrounding. Many cells, including most prokaryote, except animal cells, also have a strong supporting layer around the membrane known as a cell wall. So, in addition to a membrane, there's also a cell wall for some cells, except for animal cells. The main function of a cell wall, like the cell membrane, is to provide support and protection for the cell. Many prokaryotes, plants, algae, and fungi, have cell walls. Animal cells do not have cell walls. Cell walls lie outside the cell membrane and are porous enough to allow water, oxygen, carbon dioxide, and certain other substances to pass through easily. Many lipids have oily fatty acid chains attached to chemical groups that interact strongly with water. The fatty acids portion of such a lipid are hydrophobic or water hating, while the opposite end of the molecule is hydrophilic or water loving. This should say hydrophobic tail. When such lipids are mixed with water, their hydrophobic fatty acid tails cluster together while their hydrophilic heads are attached to water. A lipid bilayer is the result. This is the lipid bilayer. So the hydrophilic head, loving water, and the hydrophobic, should say phobic, tail. The head groups of lipids in a bilayer are exposed to water, while the fatty acid tails form an oily layer inside the membrane from which water is excluded. So there's no water inside. Most cell membranes contain protein molecules that are embedded in the lipid bilayer. Carbohydrate molecules are attached to many of these proteins. Because the proteins embedded in the lipid bilayer can move around or float among the lipids, and because so many different kinds of molecules make up the cell membrane, scientists describe the cell membrane as a fluid mosaic. So these proteins can move within the wall and move around like a fluid mosaic. Some of the proteins form channels or pumps that help to move material across the cell membrane. Many of the carbohydrate molecules act like chemical identification cards, allowing individual cells to identify one another. The fluid mosaic model is this. The carbohydrate chains are attached in here and they can move. And here's the lipid bilayer. And these are the lipids. Water's on the outside. And here's the hydrophilic polar head. And this is the hydrophobic non-polar tails. Although many substances can cross biological membranes, some are too large or too strongly charged to cross the lipid bilayer. If a substance is able to cross a membrane, the membrane is said to be permeable to it. A membrane is impermeable to substances that cannot pass across it. Most biological membranes are selectively permeable, meaning that some substances can pass across them and others cannot. Selectively permeable membranes are also called semi-permeable membranes.